thank you for the invitation to give this lecture. As I was putting the talk together, I decided to slightly change my title to the past, present, and future of genetics and evolution. We'll talk about the past, present, and future, and I'll try to remember to mention to you when I'm moving from past to present and present to future. We'll of course start in the past, introduce you to the two giants in the field of genetics and evolution. This is Gregor Mendel. He was active, or he was alive between 1822 and 1884. He discovered the fundamental laws of genetics using pea plants. His laws are applicable to all sexually reproducing organisms. His main paper was experiments on plant hybrids in 1866. So he unfortunately was not recognized, the significance of his work was not recognized in his own lifetime. It wasn't until about 30 years later that his laws were rediscovered by others, but then it was figured out that, wait a minute, this guy had it all figured out 30 years before. So unfortunately for Mendel, but he is now recognized as the father of modern genetics. The father of evolutionary theory, you all know, is Charles Darwin. So he was alive from 1809 to 1882. So he proposed the theory of evolution by natural selection. He wrote a book on the origin of species. It was published in 1859, as well as several other books, but that was the big one that put forth the theory of evolution by natural selection. All right, so let's just review let's review this theory of evolution by natural selection. So variation originates via random mutation during mitoses, and especially in the mitoses, the series of mitoses that lead to the production of germ cells. So in order for evolution to act, we must have variation, and so this is where it originates. So if you have members of a species and they vary slightly, or I guess they could vary greatly in some particular characteristic, depending on the environment, it is going to select for the most well-adapted individuals of that species. And those individuals that are most well-adapted preferentially lead more offspring, and those offspring will inherit both the adaptations of their parents as well as the genes of their parents. I should, a brief small technical detail is that the word genes is not actually the correct word. The correct word is allele. So we pretty much all have the same genes, as well as in any individual species, all has the same genes. We just vary in the particular alleles of those genes that we have. So alleles are small variations, typically small variations in a gene, like a single nucleotide polymorphism. Humans differ amongst each other at approximately 1 in 1,000 nucleotides within the genome. So we're different about 1 in 1,000 nucleotides, and that's why the person sitting on one side of you doesn't look like the person sitting on the other side of you. So that 1/1000th of the difference in the genome is why the difference, variation in humans. All right, so let's try to put the evolution of life on Earth into some perspective here. And so this diagram starts with the origin of the Earth approximately 4.6 billion years ago. Surprisingly, life is believed to have arisen on Earth 3.7 billion years ago. So that was a very long time ago, and not all that long after the Earth was originally formed. And so here we see the first life about 3.7 billion years ago. And so then for about 1 billion years, we had nothing but microbial life. So then about 2 billion years ago, the first eukaryotes evolved. So these are single-cell organisms that have their nucleus enclosed in a membrane. So those first eukaryotes were around for about another billion years before we evolved the first metazoans. Metazoans being multicellular organisms. So that was about a billion years ago. And so then over the last billion years, we have developed all of these multicellular organisms, including like the dinosaurs. I guess we have fish here about 400 million years ago, and the dinosaurs, and then we had the asteroid about 65 million years ago. And then what I want to draw your attention to right here at the very end of this timeline of the existence of the Earth and life on the Earth is where modern humans first came into being. So this was about 150,000 to 200,000 years ago was the first human. So I hope this gives you some perspective on where we fit into this timetable of life on Earth. So during this 3.7 billion years of life on Earth, we have had evolution by natural selection. So that's the way it has worked up until quite recently. I should also say, if anybody has any questions, I should have said at the very beginning, if anybody has any questions, feel free to interrupt me. I'm happy to take questions during the talk. All right, so let's take a look at the evolution of one particular structure, the eye. So when Darwin first proposed his theory of natural selection, there was a lot of opposition to it, and one of the main lines of attack was that an eye could never have evolved by natural selection because an eye is so complicated it had to arise all at once. And so that was the theory of intelligent design. What was not known at the time, nobody knew anything about DNA or mutations. They also didn't know, they weren't even close about the length of the history of the life on Earth. You know, how long had the Earth been here and how long had life evolved. I think it was maybe, they were thinking a few 100,000 to a couple million years was kind of the thinking, which is too short of a time to evolve something like an eye. So let's take a quick look at the evolution of the eye. So the primordial eye is believed to be like something that we see in today's flatworms. So you have little eyes here. It's just more of like an eye spot, really. And the vision of these flatworms is not particularly good. They can kind of sense changes in the intensity of light. They don't, they can't focus really well. They can just see changes in intensity, but it's quite poor vision. And then I show you three different modern eyes, and vision is believed to have evolved multiple times during evolution, but here we have a human eye, of course. We have the eye of a mollusk, squid, and then we have this little bizarre-looking insect with the eyes out here on these stalks. So eye evolution is highly divergent, but all of these eyes see pretty well. With the known length of time that eyes have had to evolve, and now that we understand how mutations work, this strongly refutes this idea of intelligent design. And the last point I want to make with this slide is the saying that I have at the bottom. So this, to me, encapsulates how evolution works. So the saying is, the one-eyed man is king in the land of the blind. And so what that is getting at is that your competitiveness against your fellow creatures or fellow members of your species is relative to how good those other members of your species are at what they do. So with the idea with the eye, if you can imagine some flatworms that had no vision, And then you have some that have just the ever so slightest amount of ability to discern differences in intensity. You could imagine that they are more able to escape predators than the ones who couldn't see at all, however slight that advantage might be. And then over, like a billion years of time, with all of these mutations that have an opportunity to accumulate over that amount of time, then you could develop into a mature eye that can see, with this high acuity, just amazing how well we can see. So I'll get back to this one-eyed man is king in the land of the blind statement later in the talk. All right, so this is how evolution has worked through natural selection. I would also mention evolution through artificial selection. So evolution through artificial selection means that humans have been intervening and selectively breeding organisms with characteristics that are desirable to human beings. And so with the dog species that we're all familiar with, all dog species evolved from the wolf. I see different reports in the literature sometimes, but I think somewhere in the neighborhood of 20 to 30,000 years ago, humans began artificially selecting wolves, and much more recently selecting wolves to evolving them by artificial selection in the various dog breeds that we see today. Another example of artificial selection I show you here with corn. So the ancient corn plant here is this teosinte. So this is what evolved through natural selection. And then over many, many generations of artificial selection by humans, it evolved into intermediate like this. And then this is much more familiar to you, the modern corn. But this is what it looked like when humans began intervening with artificial selection. All right, so this, I think, finishes the past part of my talk. So I'm going to move on to the current state of genetics and evolution. So I know you guys have been reading the Crack in Creation. So we're going to talk about CRISPR here momentarily and about the ethics of that. But let me just tell you where things stand with the current state of reproductive possibilities with humans. And so, of course, we reproduce the natural way most of the time, but some of the time we reproduce by assisted reproductive technology, it's called, and one of those involves this method of pre-implantation genetic diagnoses. So what is done is you have artificial insemination of a female's egg cells in culture with the partner's sperm cells, and those eggs that are fertilized are then grown in culture for several generations until there's, I don't know, maybe 8 or 16 or 32 cells, somewhere in that neighborhood of cells. And then a technician can go in and grab a hold of that fertilized embryo and can hold onto it with a suction pipette and then take a different suction pipette and pull off one cell. And the embryo is smart enough to recognize that there's a cell missing, and there will be an extra cell division to replace it. So obviously the children that are born using this method don't get born with like the absence of an arm or something like that. They turn out perfectly normal. But what is done with that egg cell that is plucked off of that embryo is subjected to genetic testing. And people use this technique to try to avoid genetic diseases, is one of the main uses of it. So if you, for example, are a carrier for cystic fibrosis, and your partner is a carrier for cystic fibrosis, if you have a child, there's a 1 out of 4 chance that that child will get both of the mutant copies of the cystic fibrosis gene from each parent, and then it will experience the symptoms, the terrible symptoms of cystic fibrosis. So I think that's pretty much everyone would like to avoid that. So if you do the pre-implantation genetic diagnosis, you say you fertilize, I'm not exactly sure, I'm not a fruit fly geneticist, but let's say you fertilize like 10 of these embryos in culture, and then you do this procedure on all 10 of them, you would expect maybe 2 or 3 of them to be homozygous mutant for cystic fibrosis, so you wouldn't want to implant those. So as shown here, don't use these ones for transplantation into the womb, but use these ones. And so using this method, we can avoid having offspring that have genetic diseases like cystic fibrosis. I'm not exactly sure how extensive these tests are, but another classic example is Huntington's disease, Tay-Sachs disease. Like I said, I don't know how extensive it is, but you could also test for Down syndrome and some other chromosomal abnormalities. But this is the current state of the possibilities with respect to artificial reproductive technologies. So there was a recent book, I'll show you the cover here, called The End of Sex and the Future of Human Reproduction by Hank Greeley. He predicts in this book that within the next generation or two that we will no longer be reproducing the natural way, that the way we will reproduce is through in vitro fertilization and then the pre-implantation genetic diagnosis. Currently, about 2% of the births in the U.S. occur by assisted reproductive technology, and that number for about the last, I think, maybe 40-ish years has been slowly increasing. Question?
Does that mean that the cost of assisted reproductive health is going to go down in the next few years?
For something like in vitro fertilization or this, I can't exactly predict when it might happen, but yeah, whenever a technology first becomes available, it's always super expensive, and there are always the ethical issues of only the most wealthy people, the top members of society, can access this technology, and the rest of us can't. But over time, it always happens that the costs go down. I'm not sure I would say the next few years, but a generation from now, I'd say it should be cheaper. I believe like one round of in vitro fertilization for a couple, I think that's just in vitro fertilization. This pre-implantation genetic diagnosis is probably more. But one of the reasons you do the in vitro fertilization if you're an infertile couple, right? And then another reason you might do it, but the infertility, you don't have to do the you don't have to do the genetic diagnosis. You just create the fertilized egg and then implant it. But if you're at risk for genetic disease, I think you'd want to also do the pre-implantation genetic diagnosis. Question?
So, like, say you have a kid with like 2 carriers of cystic fibrosis.
Speak up a little bit. I'm old and I can't hear anymore.
We're good. So is there a way to avoid, so if 2 people get together with like cystic fibrosis?
You're heterozygous, so you're a carrier
gene, not the hormone. Is there a way through genetic, I forgot what is the other word, but through the thing you just showed us, is there a way to avoid that, or is there like avoid the passing of the gene at all? If
Yeah.
Two carriers.
Yeah.
Is that what you? I think I understand.
Yeah. It's just really simple have CF, I'm going to say plus is for the normal gene, the wild type. CF minus would be the mutant. So if you have two parents, they're both carriers. These are the genotypes of the germ cells that they would produce. So you could have CF plus, CF plus, CF plus, CF minus, CF plus, CF minus, CF minus, CF minus. So this child here would be the one that would get the cystic fibrosis disease, and that would have symptoms, and this one would be affected, and probably eventually die of lung infection, probably in the neighborhood of 30 to 40 years of age. None of these three genotypes would be affected. They would not show any symptoms of cystic fibrosis because they have at least one wild-type copy of the gene. But if you pick this embryo, it is wild-type over wild-type, this person is no longer a carrier, and then they would not have the potential to pass on this gene. So you could eliminate the possibility of cystic fibrosis in your family by doing that. Right? Is that the question you're asking?
Yes.
Yeah, so you can. I think I did mention that the cost of in vitro fertilization these days, I think one round of it is somewhere in the neighborhood of like $20,000. But I think it's considerably more for the pre-implantation genetic diagnosis. So pretty expensive currently. Other questions? All right. Having trouble with this video. So you read the book about the book, The Cracking Creation, is all about CRISPR and the implications. So let's take a look here and see exactly what CRISPR is. So this movie's about 4 minutes and something long, but I think we're going to look at the first 2 minutes and then I'll cut it off. Is it going to play? I've just got so much trouble with this video. I double click it. Darn, not going to play. But let me describe for you what CRISPR is then. So CRISPR is, there's two components to CRISPR. One is a protein, it's a nuclease, which will cleave DNA. The other part of it is it's called a guide RNA. So the CRISPR method, you hear a lot of hype about it, but all it really is the ability to make a targeted double-strand break in a chromosome. That's all it really is. And in that guide RNA is a slight variant of DNA, but it has similar base pair sequences. You can pick about 20 nucleotide sequence in that guide RNA, and then that guide RNA will search through the entire genome. It will find the 20 base pairs to which it shows matching nucleotide pairs, and it will take that nuclease to that particular spot in the genome, and then that nuclease, the protein nuclease, will cleave the chromosome right at that specific spot. So it's like these molecular scissors that have this ability to precisely target it. And so you can put any 20 base pairs you want into that guide RNA and cause the Cas9 protein to go anywhere you want it to in the genome and make that double-stranded break. And so once that break has been made, the chromosome will try to repair itself, and sometimes, a lot of times, it makes errors. It will put itself back together, but it will have deleted or inserted some base pairs, and you will have mutated the gene that's where the location of where the cutting was. But there's an additional possibility of adding a third component. It's called a donor plasmid, but it has certain sequences that will insert into the spot where the double-stranded break occurs. So you can engineer a gene pretty much in any way you want it if you add this additional component, this donor plasmid with the sequences of DNA that you want to be encoded in there. Sorry that video didn't work. I put it in there and it doesn't work. And I just put it in there like an hour ago and I thought for sure it would work. Oh, it's going to work.
Cas9 system is a tool for cutting DNA at a specifically targeted location.
You guys can't hear this, can you?
The technique has
All right.
already revolutionized gene editing, but scientists are always looking for new possibilities. So what else
We could try to remove the closed captioning.
Can't seem to pause it. Say that again?
Since the introduction of bacterial diseases, CRISPR-Cas9 has been adapted.
I'll just try to narrate. So the Cas9 is the protein there, and the red thing is the guide RNA.
And an RNA molecule known as the guide RNA. Altogether, they form a complex that can identify and cut specific sections of DNA. First, Cas9.
So that's the chromosome there. So that's the guide RNA in red. See the specific base pairs in the guide RNA.
Found, the guide RNA unwinds part of the double helix. RNA strand is designed to match and bind a particular sequence in DNA.
So that's very specific of one particular spot in the whole genome.
Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
All right, so I think that's what I wanted to show you. So that's the basic process of how CRISPR works. So typically you just need two components. So it's relatively simple, guide RNA and the Cas9 nuclease. I would also say it's been possible to modify the genome for about 30 years, but the methods prior to CRISPR were very difficult and quite expensive, and so it just wasn't commonly done. But the word that's used with CRISPR is that it has democratized the ability to genetically target, to do this genome engineering in this targeted way because it's relatively easy and quite cheap. Question?
So the way basically that CRISPR works is that like it knocks out a gene, so the desired gene that you that hasn't shown itself before will take its place and express itself.
Well, if your goal is to knock out a gene, all you have to do is put that specific sequence of the gene into your guide RNA and then add the Cas9 and then it will go in and it will cut. And then it's kind of a statistical thing. Sometimes it will repair the chromosome right back to normal, but like it showed in the video here, it added a few nucleotides, and so it made a mutation in that gene. And so you have to screen like individual embryos to figure out which ones got repaired normally and which ones got mutated the gene that you were trying to target.
So how do you initially put the Cas9 protein on the desired chromosome? Are you doing that, or does the Cas9 know how to find the right chromosome?
So if you are going to, I've been doing CRISPRing, doing the genome editing of fruit flies for like 6 or 7 years now. But people do it in mouse, and the mouse method would be much more similar to humans. So what you would do is you would fertilize some mouse embryos, and right at that one cell stage, so before you have any cell divisions at all, you have a single cell zygote. It's a diploid because both the sperm and the egg came together, so you have both parents' chromosomes are there. You would then inject that, right? You've got to have these outside the body of the mouse at this point. You would have in a petri dish with a suction pipette holding onto one, and you would have an injection pipette that you would physically inject the guide RNA and the Cas9 into that egg cell, and then the magic that we just saw in the video would happen. But as I was saying, if you just want to knock out the gene, you just need the guide RNA and the Cas9. But if you want to put some specific sequences in there, you want to modify that gene in a particular way, you can also add this extra piece of DNA, the donor plasmid, and then the donor plasmid, a high percentage of the time, the repairer will use the donor plasmid to put in different sequences, whatever sequences you might have wanted to repair. so if your ambition was to be the first person to do genome editing of the human germline using CRISPR, you are too late. You're going to have to find another Mount Everest to climb. So in 2018, a Chinese researcher reported that he had germline genome edited a pair of, I think they were twins, some Chinese twins that were female girls, and what he did was he knocked out a gene called CCR5, and it has been known through the study of HIV infection that some people that you would have expected would have got infected with HIV did not, and it was determined that the ones that were at high risk but did not get infected had mutations in this gene CCR5. So he knocked out this gene, and his rationale was that he wanted to make these children resistant to infection by HIV, and he probably did that based on what we know about this gene. But he did not get any, for any sort of work like this, there's all kinds of ethical review boards, and a whole lot of people have to be involved, and everything has to be approved. He did none of that. He just went ahead and did it without any regulatory oversight whatsoever. He was convicted of breaking the rules. He did 3 years in prison, and just a few months ago, I was reading up, he finished his 3-year prison sentence. So this is the one example that we know of where somebody has already used CRISPR to do the genome editing of a human. I don't know any other examples. I don't think any other examples are known, but it wouldn't shock me if it's happened another time. I would also point out that CRISPR, sometimes CRISPR gets compared to nuclear bomb. All right, we got the nuclear bomb back in the 1940s, and within a year or less of developing the atomic bomb, we used it on other people to kill other people. It was a huge government project, a lot of really smart physicists involved, and huge government funding to develop the atomic bomb. A difference with CRISPR is that it's relatively cheap, and you don't have to have a whole lot of super smart people involved. So, if we decided in this classroom that let's go make an atomic bomb for our project for the semester, we'd never be able to pull that off, right? We don't have the funding, we don't have the know-how, but I don't know how to do exactly the injections of the embryos, but I could probably, of the mammalian embryos, but I could probably guide us pretty easily to doing that. But probably you just need 2 or 3 people and a couple of relatively low power microscopes, probably for a few $1,000, maybe $5,000, $10,000 at the most, you could do this. So CRISPR is much more easily done than atomic bomb. So why is this such a bad thing that he did? Well, there's this genetic concept called pleiotropy, which is that a lot of genes don't just do one thing, they do multiple things. So we know that this CCR5 gene is involved in susceptibility to HIV infection, and like I said, he probably has made these twins immune to infection by HIV, but a lot of genes, they do more than one thing. They might affect, in this slide, we're showing trait A, trait B, and trait C. So if he gave HIV infection resistance by trait A, what trait B and trait C does this gene do? So what other problems might these twins have? We just don't know. One thing I would say the geneticists are really bad at is predicting what the phenotype will be if you knock out a particular gene. It's like, I want to knock out gene X. Okay, let's knock out gene X. What did you predict would happen? Well, the phenotype that you get is usually not what you expected. All right. So, how do we decide whether we should use CRISPR to manipulate the human genome? So let me just give you a classic example of basic logic. Can we use deductive logic to decide whether we should use CRISPR to genome edit humans? So let's take a look at this example here. So this is a classic example. All unmarried men are bachelors. Bill is an unmarried man. If you accept premise 1 and you accept premise 2, then it must be the case that 3 is true. Bill is a bachelor. This is kind of like 2 plus 2 is 4. 2 plus 2 is 4. It doesn't matter what universe you live in. It doesn't matter what your skin color is, what your religion is, what your anything about you, it's going to hold. All right, so let's take a look at a different set of statements here. So, genetic disease causes human suffering. I think we can all agree that that's the case. 2, CRISPR can be used to cure genetic disease. Certainly for some genetic diseases that is also true. So 3, humans ought to use CRISPR to cure genetic disease. So is this fit the pattern of what I showed you in the previous slide?
It has a keyword can, which indicates that it might not work.
I mean, do you disagree with the statement that CRISPR can be used to cure genetic disease?
No.
No? Okay. So we all agree on 1 and 2, right? Do we agree that 3 logically follows? If 1 and 2 are true, do we agree that 3 must be true? Yes? No?
Logically, yes. But that doesn't mean there's other factors that come into play.
What if, yeah, so that, I think that's the key thing, is that there could be extra information that would make you make a different decision. So if I told you that CRISPR costs a lot, which actually doesn't cost a whole bunch, but let's say it does for the sake of argument, and let's say we had to divert resources from some other government program. Let's say a bunch of children have to starve in order for us to use those resources to cure these genetic diseases. Well, that might make you make a different decision, right? David Hume was the first one to point out to us that you cannot deduce normative ought statements from descriptive is statements. So no matter how many factual statements about the world there may be that you could use to support your claim, you cannot, does not logically follow that you ought to do something. So found this little slide on the internet. It's called Hume's guillotine. So if you have the world of facts, a bunch of is statements, you cannot derive ought statements or normative statements, value statements about the world. So we can't use logic to deduce whether we ought to use CRISPR to manipulate the germlines of humans. I mean, it's a super tricky question, all right? So maybe we could use religion to guide our ethics, but there's a lot of different religions in the world, and not everybody's going to have the same ethical framework. Not everybody's religious. Maybe they have a non-religious, secular sort of framework. So it's no clear answers whatsoever. And there have been arguments all over the map as to whether we should use CRISPR. Some people have argued that absolutely not. We should just stay away from doing that, at least for a long time until we're much more scientifically advanced. Other people have argued that there's human suffering occurring because of human genetic diseases. It's morally wrong not to use CRISPR to try to cure human genetic diseases, to alleviate the suffering of those individuals. All right, so I made up a little list here. It's certainly not a complete list of possible uses of CRISPR for good. So as we've just been talking about, cure human genetic disease. So CRISPR has been used and is currently being used to treat genetic diseases, but this is manipulation of somatic cells. So this works especially well with cells of like the blood. So you can take cells out of a patient that have a certain genetic defect. You could, in culture, you could use the CRISPR to correct a genetic defect, and then you could put the genome modified cells back into the person, and they would be much better off. I know that it's also been used for certain eye diseases. As painful as this sounds, you can inject into the eye, and with like a modified virus that will affect the photoreceptor cells, and it can go in there and change a mutated gene back to the normal or wild type version. So the eye is relatively accessible, but if your option is to be blind or get an occasional injection into the eye, I think most of us would go with the injection into the eye. Other uses, agriculture, both plants and animals, increase the yield of crops, grow animals with desirable characteristics. I kind of skipped over, I should have said a little more here. But with the germline, with this somatic manipulation that I was just talking about, that person that underwent the somatic treatment cannot pass it on to their offspring. So the ethical line that we've drawn as humans is that somatic cell genetic manipulation is okay, but germline manipulation, germlining meaning that which can be passed on to future generations, is not okay, at least at present. You also can imagine using it to fight communicable diseases. Laboratory research, that's what I do. It's been CRISPR has been tremendously useful in laboratory research, ability to do exactly the genetic change that you want relatively easily. You might be able to bring back extinct species. There's talk about bringing back the woolly mammoth. We could also use it for warfare. So if you could imagine current villain on the world stage is Putin, right? He's over there in that war in Ukraine. His armies or soldiers are committing atrocities. So maybe we could genetically engineer something that would kill those soldiers, and we could prevent that war. So these are just certainly not comprehensive, but one list of CRISPR for good. So here's my list of CRISPR for evil, and you'll notice that it's exactly the same list on the previous slide, but gone wrong. So for curing the human genetic diseases, I mentioned the pleiotropy, you try to do one thing, but some unintended consequence happens, and maybe you help a person in one way, but maybe you harm them in another. What if we created some super plant that starts to take over the world, or some animal that we can't control, trying to change communicable diseases? We think we're going to change it in a way that will help people, but it makes it worse. Who knows what could go wrong with some laboratory research. Bring back extinct species that we all seen Jurassic Park. That didn't work out so well. Maybe it's great if we can think about using warfare to stop the Russian soldiers in Ukraine, but what if some other country wanted to use it to do something bad to us? So every one of these is potentially subject to bad consequences. So you've all heard of eugenics. When we talk about eugenics, we mostly talk about Hitler was trying to use eugenics. Question?
Back to the previous slide, could you elaborate on how the extinct species could be brought back?
So they are, especially with like global warming, I know that there have been a number of fossils, and not actually, they're not really fossils, but like the woolly mammoth, right? So we have the complete sequence of the woolly mammoth genome at this point. And, the woolly mammoth and the elephant are relatively closely related. So we know exactly, like every DNA base change is different between an elephant and a woolly mammoth. And I don't know why, somebody seems to be really into this, maybe some billionaire is funding this project or something, but you can imagine the repetitive use of CRISPR to go into the elephant genome and keep changing those base pairs. I mean, it seems like it'd be unimaginably long and complex and difficult, but in theory, you could change every one of those base pairs in the current elephant to the one and get an elephant to incubate the embryo once you have it and you have a woolly mammoth. So, yeah. All right. So, what is the pitfalls of using CRISPR for to manipulate the germlines of humans? So, as I was saying, we all know that Hitler had like a eugenics program, but Hitler wasn't the only one. As Americans, we want to try to gloss over our own thinking on eugenics. So eugenics, if you're not aware, was programs back in the early part of the 20th century, like 1930s, 40s, even 50s, I think. It was, we want to not have the people at the lower end of society breeding. The people who were poor, maybe criminals, stopped those people from breeding. There were some sterilization programs, but it's been universally, or nearly universally, decided that that's not the way to go. Who knows? Maybe certain people, right? We have lots of poor people in this world who've gone on to do great things. And so if you try to eliminate certain genes from the gene pool, maybe you eliminate a potential future Einstein or a potential future Mozart. Who knows? But eugenics has a very bad reputation. But with this CRISPR, it is possible to kind of get back to that sort of thing. Is what sort of characteristics do we want in humans? So now I'm moving on to the future part of the lecture. So here's another book by the same guy called CRISPR People, Science of Ethics of Editing Humans. So this is now, up until now, we've had evolution by natural selection, and maybe a little bit of evolution by artificial selections of humans selecting variation that already existed, that was created naturally. But now we have the possibility to do evolution by intelligent design. And there's just all kinds of ethical questions that come up with this. So let me ask, raise of hands, is who gave consent to their parents to be conceived? Anyone? No, it's pretty much impossible, right? So one problem with evolution by intelligent design is that you don't get to decide, right? So now we can do this. But parents, or maybe the state, has to decide ahead of time, right? And then the individual gets born and they're genetically engineered. But they have no say in it, right? So the person that's affected is not the person that had any volition or consent to being genome edited. I would say over a long time frame, right, I showed you that long path of history of life on Earth, and humans are that tiny little, I mean, just imagine, 100,000, 10,000, a million years from now, I will say that there's inevitably humans will genome edit ourselves. Sort of things that would be worrisome that people might have to think about is, what if the Joneses down the street are genome editing their children and trying to give them genetic enhancements? And well, should I genome edit my children? And what if they're going to be super smart children and very athletic and really good looking or whatever? So just to keep up, you have to do this just to keep up with the Joneses, right? I'm not saying this is going to happen tomorrow. It's probably not going to be your problem, but maybe 2 or 3 or 4 generations from now, these might be things we have to think about. And also like warfare. What if Putin is over there genome editing, creating some super soldiers that are super stronger, ethically, absence of ethics. They have no empathy. They've been genome edited to have no empathy for other human beings, right? That'd be a great characteristic for a soldier. What if he's doing that? Should we start doing that to create something that would counterbalance him? So really sticky, thorny evolutionary questions. I would mention to you a couple of things that are always cited whenever we talk about genome editing and eugenics. One is this novel Brave New World. It's relatively short, all the way back in 1932. It's a dystopian novel about what if we have societal classes based on genetics and controlled by the state. So in this dystopian novel, he has the alpha, beta, gamma, deltas, and epsilons, and so the state decides how many alphas do we need. They get the top jobs in this society, the betas get the next tier, and so on. The epsilons get the worst. There's actually a series I noticed on Peacock. I watched a couple episodes. It was pretty interesting. It doesn't try to tell the tale of the book, but it uses the premise of the book and it tries to tell a different tale. So you might find it on Peacock. Another movie that I would, or this is a movie that I would recommend. So this was 1997. This movie is called Gattaca. In this dystopian movie, individuals, basically you do the pre-implantation genetic diagnosis, and the couple looks at all the embryos. They say they have a dozen embryos or whatever. They look at the genetic profile, all dozen of them, and then they implant the one that the couple chooses, and they want to have the best gene. So right now our pre-implantation genetic diagnosis is only to try to eliminate genetic diseases, but in this situation, they're trying to look for genetic enhancements. And basically your genetic profile determines what careers are open to you, what positions in society are open to you. So this is a movie I highly recommend. So I guess if I only get across one message in this talk today, I want to get across this idea that CRISPR is a real Pandora's box. So Pandora, this goes back to Greek mythology. Pandora was a mortal woman. Zeus gave her this box, and he said, the contents of this box is not for mortal eyes. She put it off for a while, but eventually her curiosity got the better of her. She opened it up, and you see all these demons going out into the world to cause who knows what kind of mayhem. And so Zeus had been putting the like souls of all the demons in the world into this box, and she released it. So if we start genome editing, especially humans, it's just a real Pandora's box. It's just really hard to know, even if we have the best of intentions, what sort of unintended consequences may result. So my last slide, bring it back to here. So up until this point of the history of life on Earth, we've had evolution by natural selection, and then somewhere about here we had evolution by artificial selection for a few species like dogs and some agricultural plants, maybe our agricultural animals. But now, at this point, going forward, we're going to also have some evolution by intelligent design. So this opens a new era. So I talked about like intelligent design, right, was a theory that was used to try to refute Darwin's theory of evolution by natural selection. It's largely discounted among any serious scientists, but now we actually do have the potential to have evolution through intelligent design using CRISPR. I'm just out of time, so that's it for the lecture. Anybody have any questions?
Do you think Pandora's box has already been opened?
Well, I mean, I mentioned, a little bit ago that CRISPR is too easy and too cheap. So it just takes 2 or 3 people cooperating and a few $1,000s to do some sort of a genome editing on people. So yeah, there's been a laws passed. It's no germline editing of humans. That's the ethical law. There's the ethical rule. There's laws against it. You go to jail if you do it currently. The technology is out there. It seems like maybe there ought to be a little more tighter regulation on it than currently is. But it's hard to say. But probably the answer is yes. Yes, I would say.
How long do you think till we figure out what every gene does exactly?
Yeah, so see, that's the sort of thing. Like, let's say we wanted to make, let's say you were determined to make your child higher intelligence than they otherwise would be. At the moment, I don't think that even if you were given the money and it was ethically approved to do that, it's not known. What gene would you go in and mutate? How would you change what gene? It's not really known. I think we're accumulating evidence of data on genes, and it's only going to accelerate ever faster. But I think there will come a day when we do know what genes we could change in order to make humans more intelligent, and then it will be an option to do it. I would say decades. You said like everything that every single gene is doing. Maybe never, but we will progressively and gradually accumulate knowledge about different genes and how we can modify this gene or that to cause this or that phenotypic effect, and we will get better and better at that over time.
For diseases like cystic fibrosis, do you think it's within the realm of possibility to have someone be born with cystic fibrosis, and then even though they already have trillions of cells in their body, go in with CRISPR and change every individual cells until they no longer have cystic fibrosis?
It's just super hard to do that. People have been trying, right? Cystic fibrosis gene was cloned 40 years ago, maybe 43 years ago, actually. And the treatments for being able to go in there, we should be able to go in there and correct it. And the lungs is the most affected organ that usually causes lung infections that cause death. But it's relatively minimal what we've been able to do with any sort of genetic changes. So the problem is, that's why the change needs to be done at the single cell stage, right? Because then every cell that divides after that has that change. How do you get the CRISPR into the trillion cells? It's just cells that are external, cells in the blood, those ones we have access to. How do you get to the cells, in the brain or deep in the body that are many, many, many cell layers deep? It's just a practical problem that people have been working on this for decades using viruses to try to do these infections, but it's really hard to get more than that first layer of accessible cells. So not easy, not soon. Yeah.
To execute what he was suggesting, would you have to edit every single cell that contained that gene, or a majority or a fraction of them? Would it cascade in effect?
So with cystic fibrosis, it encodes a chloride channel, and it is particularly, the phenotypic effects are felt in secretory glands. So the lungs is one, even though they're not particularly secretory, but there's secretory tissues like the pancreas and some other secretory tissues. So you really only need to do the correction in the cells that are having a phenotypic effect. So no, you wouldn't have to correct every cell, just the ones in which that gene is known to function.
And would it be all the cells within those tissues tissues that would exhibit that phenotypic effect?
I think if you could even get part of them to have the genetic correction, you would see like a reduction in the phenotype. So the more that you could get the correction into, the less severe would be the phenotype. Yep.
If we, again, for cystic fibrosis, say that we collectively decided to basically do, I would call it maybe like a genetic vaccine against cystic fibrosis, meaning that everybody got an implantation. We had one generation where we just said everybody ends up with the double CF plus, so that there is no allele for cystic fibrosis. Would that eradicate cystic fibrosis, or what are the chances that because it's already mutated into a thing, that we would just end up mutating back to a cystic fibrosis?
I mean, if you looked at enough people over enough timeline, you would get you would get some more mutations in it, but you could pretty much effectively cure it and eliminate it from the human gene pool. But we don't have to use CRISPR to do this, right? We just have to do the pre-implantation genetic diagnosis. You don't need CRISPR to do that. If you just screened everybody that's a carrier or anybody that's affected, just keep doing the pre-implantation genetic diagnosis, keep selecting for the CF plus. You don't need CRISPR to do that. And that would be permitted within what we've decided as a society based on our current ethical guidelines. So we're just selecting something that's already there. We're not manipulating the germline at all doing that. Yep.
And what is the success rate for reimplantation? Are we seeing that they create like healthy kids at the same amount of time as a regular birth, or are there some side effects inherent to taking
I think that, again, I'm not like a human reproductive technology kind of person, but my understanding is that people that are born using assisted reproductive technologies don't have any more sort of problems than people that are born through natural fertilization. Like I said, you pluck off that one cell, it's not like, okay, that person's born without an arm now because you plucked off that cell. The cell embryo does a lot of time to develop. It knows there's a cell missing. There'll be an extra cell division to replace it. And, I don't think that there's really any negative effects really on people born through. I think it's been like 40 years or maybe even more than 40 years since the first in vitro fertilization baby was born. So I don't see any of that talked about in the papers that I read. More questions? You have a question?
Yeah, is there
Speak up a little, I'm old, remember, can't hear.
is there potential for the creation of new species through CRISPR?
Absolutely. Yeah, and one thing that has crossed my mind is taking humans and diverging us into separate species. So I think one thing in temptation, if you remember back to the Industrial Revolution, back pre-Industrial Revolution, you had an individual shop maker would make some particular product, and he would do every step from A to Z to make this product. But then the Industrial Revolution came around, and we have assembly line. So you have one person will be on the assembly line, and they'll do one step, all day, every day. So you specialize. So that enhances productivity. So you can imagine that there will be a temptation to generate humans. Could be the same, we could still be human, but we could be different species that are designed to do very specific tasks and be very good at one specific task. So, but absolutely, can create new species. Yep. And they've even been able to synthesize genomes of microbes de novo. So I guess I could say that it's already been done with bacteria. But yes, it definitely has the potential to be done using CRISPR on more complicated organisms like people or mammals and stuff. Yep.
So aging is just changes and mutations in your cells, right? And aren't they kind of programmed to age? So can we do something with CRISPR to change, or like other technologies to change that so we don't age as fast?
I think that certainly potential is there. Yeah, like you get a shortening of your telomeres. Like every time your cells divide, you shorten your telomeres. That's one of the effects of aging. Your telomeres get reset when you're during the germ cell development process, and so you just reset at birth. But yeah, absolutely, there's a potential to adopt CRISPR to alter the genetic changes that occur with the aging process. So that would create a whole huge, another ethical problem if we, I mean, we already have, I think, enough overpopulation in this world, but what if people live 10 times longer? What if we live to 1,000, instead of about 100, how many people we have on the planet? So, yeah. Anything else? Good. All right. Oh, more questions?
Is there the potential for the body to adapt against the mechanisms of CRISPR? Because like a single alteration in the structure of the actual genetic scissors can like completely change its function on the no?
I don't really see it. I mean, if there was some adaptation, we're smart enough that we could get around it. So I mean, the way that you would genome edit a human is you would in vitro fertilize the human egg with a sperm. And so you got this single cell diploid zygote, it's a single cell. And then you hold it with a suction pipette and you inject your Cas9, your guide RNA with the injection pipette. So I don't know how you could develop any sort of resistance to that. I mean, this is not like a natural process, right? This is, humans are very actively intervening here. So I don't think so.
What would the process look like to create a new species with CRISPR?
I mean, it's not very feasible
Would it
at the moment to do it, but like, I mean, let's say, tell me what sort of species that you would want that doesn't currently exist. Like what sort of combinations of features of other species?
I don't know. I would have to think on that for a while.
I mean, the way that you change like the structure of a body is not usually by like creating whole new genes that then work together to create that structure. The way that evolution typically creates a new structure, like an example I might give you would be like the wing of a bat relative to like a leg of an ancestor. So there's not like a whole bunch of new genes that are evolved to create that wing of that bat. What happens is you change the timing or the amount or the location of genes that already exist. So evolution tends to be lazy. So you would be mucking with the regulatory regions of the DNA that tell genes when and where and how much to be expressed, and that's how you change a normal limb of an ancestor into, say, the wing of a bat. So, but yeah, I mean, you could just, I don't know, certain characteristics from different species. The rules of genetics, you got to have matching pairs of chromosomes, you got to have the same number from the male parent versus the female parent, and so on. But yeah.
What's your current research focus with fruit flies?
I studied neurobiology of Drosophila. Neuroscience. I would say that, if I didn't say it already, that I can see the Gattaca situation being not unrealistic within a generation or two, where the parents go into the reproductive clinic and they fertilize a dozen or so embryos, and then you pick the best one. Like I said, currently we don't allow anything but selection against genetic disease, but even without any CRISPR, we could select for certain characteristics that might be desired, the parents might desire in their offspring. So give it a generation or two. I think in that end of sex book, he predicted like 30 to 40 years. So your children and their children, that sort of a Gattaca situation might be realistic, where we're not reproducing anymore by the natural sexual process, or in vitro fertilization and selecting whatever desirable conditions might already exist, right? In the Gattaca movie early on, the technician says, it's all you, these embryos are all you, there's nothing, we haven't changed anything, we're just selecting the best of you. So I think that could be an ethical dilemma, which just in a couple generations